So hello. So my name is Sebastian Kadener. I'm one of the, I would say, the main teacher of the class with Mona Soreq. And the first thing I will tell you is that if my accent is difficult for you to understand, just let me know and I can speak slower. If there is any word that you don't understand, just ask me again, okay? Okay. There's a marker. Okay, so today I'm going to mainly talk about, the title of the lecture is neurochemistry of neurodegeneration, but the idea is to give you the biological basis for what happens in broadly in what we call neurodegenerative diseases, okay? So this is going to be very general, but you are going to see that in the slides there are a lot of details. And this is not to scare you, this is just for the ones of you that wants to go deeper and look deeper, but you don't need to know the details. You don't need to know the name of the protein. You don't need to just want to put it there for those of you that want to go deeper. You need to grab the concept, but I think I will repeat the concept so many times that will be clear what is the concept. And if you have any question, again, ask me. So again, ask me. So what, can you just go very quick over the class? What is the background of each one? Very quick.
Sociology and education.
Sorry?
International relations. English and French literature. Social gerontology. International relations and East Asian studies. Psychology and education.
Economics and cognitive sciences.
Okay, okay. But you have a little of biology background, no?
Yeah.
A little, and Karen talk. So don't be scared, okay, for what you're going to see, because no, I know what is the background, but just wanted to be sure. So I just want to tell you what is, in general, why I believe I need to be standing here and not just give you 10 pages with what I'm going to say. And this has to be because I believe that the teaching and the experience of teaching and learning needs to be very interactive. And I try to do my best for this. So my leitmotif usually is this, I think this phrase of Jacques Monod is a very, illustrates very well the way I think, in general in science, not in research, not just in teaching. So in science, self-satisfaction is there. Personal self-satisfaction is the death of the scientist. Collective is the day of the research. And he said that it's restless, anxiety, dissatisfaction, agony of mind that nourish science. And so if you feel any of these things, I'm succeeding, okay? Okay. So, and also this means that basically don't be afraid to ask me. We have, I think I only teach 2 or 3 classes here. So you should really kill me with questions. There is no problem if we don't reach all the material. It's not going to harm anyone because, you know, I think these classes are important, the ones I'm teaching, to give the framework for all the guest lecturers we have. So we start with here, okay? Ah, wow. Can you read up there? No, how we do then? Because the computer is fine. It's just a shadow here. Ah, wow. Okay. Okay. So the first question we have to, I would like to pose here, if is our body a beautiful and a perfect machinery? And I remember when, since I'm in a primary school, they tell me how fabulous is the body, how beautiful is working, how well designed it is, you know. And, you know, some religious people will say that it was designed by God, and other people will say it's designed by evolution, but it's so perfect. So when I then grew up a little, and instead of taking biology classes, started to take physics classes, I started to realize that the processes in the body are very inefficient, and the body has a very, very bad design. So I see, the way I see actually the body now is it's more like a machine in the verge of malfunction. And the miracle is that keeps working, not that that is working. So then I believe that the picture we should have is like this. And at the end, unhappily, the body always collapses. Okay? So what we are trying to see in this class is what are the elements to make what I believe is the most important machine in the body, which is the brain, to collapse. So for you guys to know, the cells operate in a very narrow window of conditions. So we have our body, and you know that we are what we call a homeotherm, so our body is keeping 37 degrees. So each cell in our body, despite we are in the Antarctica or we are in the North Pole, is always facing the same temperature, okay, around 37 degrees, every cell inside our body. So in general, if you take cells and you put them at lower temperature or higher temperature, the cells are going to die very, very quickly. And this is because usually our enzymes, you know, the enzymes are the mainly proteins which make everything in cell function. They are usually selected in a way that operate much better at 37 degrees. So if you move the cell from the optimal temperature, you're going to have a much less enzymatic activity. If you increase temperature, the proteins are going to lose their shape. You know, the
Structure.
Yeah, the shape, the structure, exactly, yeah. So you know that what gives function to a protein is the structure, is the shape, the way they fold, and depending on the way they fold is what they can bind and what they can do. So if you increase temperature, the proteins are going to denature. And also many of the processes in the cell are based on some of them in diffusion, some in facilitated diffusion. If you decrease temperature, you will have many of the processes are going to happen very, very slow, too slow maybe. So not only the temperature is important, but also the ion concentrations. So the ion concentrations that the cell faces, or that the cell can live happily, let's say happily, okay, are very narrow. Also, of course, if we put a cell without nutrients, the cell is going to die. So, and this window is not very wide, and there is many other conditions. And the point here is that really the cells operate in a very, very small window of conditions. So, and we know that our body is subjected to changes in conditions all the time. And this means from the outside as well from the inside. So it's very important that we have systems that minimize these changes, or that minimize the changes that the cell feels. The best example, I think, is the temperature. Our body keeps 37 degrees. Spend a lot of energy on that, but it's a way to give the cells the right environment. So I believe for keeping the class, we need to refer to the concept of homeostasis. Basically, homeostasis is the property of a system that regulates its internal environment and tends to maintain a stable, constant condition. So basically, we say that the system is homeostatic if it can regulate the internal environment, and this internal environment is maintained constant despite changes in the external conditions. So what we can think in a funnel, and let's think like what the outside of our body is seeing is a very wide changes in temperature, in chemical stimulus, in ion concentrations, in everything that you can imagine, and somehow we have systems that make this difference down to be very, very small. Okay, it's just constant. Don't read much inside. Okay, so how you can create a system which is homeostatic, and the main principle, I think that Karen talked a little about this last week, she told me, is if you have is by feedback loops, by negative feedback loops. If you have a sensor who is going to sense a factor, who is going to sense some stimulus from the outside, this is going to be to signal to some integration center, and then we're going to have a response that inhibit this factor. The typical example, if we want to keep the let me see, the heart rate constant, so we have a way to see how many times the heart is beating every minute. If we have a sensor who is counting how many times it's beating, and this is connected to integrating center to effector, if we are below, we are going to make it faster. We are more, we are going to make it lower. And this is the general concept how many of the regulations in our body works also with temperature. So, and if I believe correctly, Karen talked a little about this last week. So the key concept then to keep until now is that the normal cells have a very narrow range of function or steady state, and we call this homeostasis. If we have too much physiologic or pathologic stress, the cells may move to a new type of state, which we call it adaptation. Okay. If we now give more stress, then the cell cannot adapt anymore, and what happens is that we have injury. So usually the adaptive situations are already situations in which there is pathology already, okay? Because we are the cells are exposed to conditions that are not good for them, and they cannot. So what everything I'm talking about has to do here. So if we look different cell types in the whole body, you need to know that the neurons are very special type of cell. And in particular, the synapses are structures which are very, very, very sensitive to stressors. We have here just one example of one known. That doesn't. I don't want to go too much into detail. You know what? You study little organelles, mitochondria, no? Yeah. You know what is so particular about a neuron? What a neuron has or is doing that you believe can be bad for it. Let's wait a little more. Yes, I think I have it afterwards. Okay. So what I'm going to say is that let's do it again. So what are the properties of the neurons? So I come back to the same that I decided to do, but I will do it. So how we can distinguish a neuron? Not based on morphology, okay? Just let's think in properties, general properties of a neuron. If you have to say something about a neuron, what is the first things you guys would say? Respect to other cells, like to a cell in the skin.
They use activation potentials.
Okay, they use electrical, they use, so if you, yeah, okay, this is true. And yeah, this is true. But this, what else with the neurons?
They're built specifically to transmit, I want to say messages, but that's not quite right, but to transmit stimuluses from one cell to the other.
Okay, they are communicated one to each other. And in order to communicate, they share connections, okay? So you know that the neurons have a lot of membrane, a lot of surface is exposed to the outside. I mean, if you have a cell, I don't have a mark here. You do? Thanks. But if you have a cell, thank you very much. If you have a normal cell, you can think in a cell like this. Like a typical cell, let's say, has like this. So you can calculate the surface that this cell, how much is exposed to the outside, is basically how much membrane, plasma membrane you have. Now, when you have a neuron, you know that the neuron, especially in many cases, you have very long axons. But let's think now what happened in the terminals. You have what is called spines, you know, and this spine you have handles. I'm very bad drawing, okay? But you have a lot of imagination and you know the picture. So you have, in each one of these spines, you have a lot of synapses, okay? A lot of synapses. So you have a lot of surface in respect to the volume. Even if you just think in a cell like this, and now if I make a neuron like this, you can easily see that the amount of membrane in respect to the amount of surface which is exposed to the outside is much more than in this case. And I'm telling you that you have the synapses forming these spines, which really have tons, 3, 4, 5 times more surface. So this means they are much exposed to the outside. So if there are stressors in intracellular spaces, they have much more membrane, they have much more communication with extracellular spaces. So just because of this, they are much more exposed to stress. This is number one. So you know that one of the main sources of stress is free radicals. We are going to talk a lot about this, but free radicals and something that happen associated with the use of the mitochondria. When you do a glycolysis, you have and Krebs chain, and you generate ATP in the mitochondria, you generate a lot of stress, which the cell has to deal with. The cell has mechanism to deal with this stress, or this we call it oxidative stress. But what happened? You know how much energy a neuron uses? It uses so much more energy than almost any single cell in the body. Okay, the brain uses a lot of sugar. I don't know if you know when you. I don't know if it's because of this, but sometimes maybe you have a headache and you eat some sugar and goes away. Okay, so the brain is the organ which uses more sugar than any other organ, and this is because it needs a lot of energy to work. A lot of energy to work. Okay, and the use of a lot of energy comes together with the generation of toxic substance like free radicals, etc., etc. Okay. What else happened with the neurons? Imagine you have I'm trying to think which I have. No, but there is another key property of the neuron, that the neuron is very different. You guys said, well, communication with other cells, presence of electrical stimulus. What else? A lot of energy, something very, very easy.
It does not degenerate.
Exactly. So the neurons don't divide. So this means if a neuron dies, it's not replaced, number one. Number two means it's like if you have damage in the neuron, year after year after year after year, what happens? The damage accumulates there. Okay? In any other cell divide, and after all the damage get the mitochondria, the membranes are made again. You have only half of the one before, but this happens. You know, most cells divide every day, every 2 days, every 3 days. You know, but the neurons, most of them don't divide, so you have it for many, many years. So the damage accumulates. And these are the three main properties of the neurons that classically people believe are associated with the neurodegeneration. So these three properties make the neuron to be special and make us to have higher functions, let's say, but also come together with they are fragile also, and these come together with this. So the age, and that's where we're going to start now. So the age conspires against the capacity of the neurons to keep homeostasis. So because of the neurons use so much energy, they have such a long or big surface, big membranes, and they're exposed to a lot of stress, and because they don't divide, for them it's very difficult to keep homeostasis. Okay? They do a huge effort every day to keep homeostasis. As the cells age, as we get older, they need to do more and more and more work. So, and I just first I want to give you the what is the canonical or what is the typical I want you to have the common view in the field, or the common view for the last 20, 30, 40 years in the field. And then I'm going to tell you that maybe it's a little different, but I want first to build this way, okay? So it's not that I'm going to contradict myself later, but it's on purpose, okay? So with age, we're going to have an increase in oxidative, metabolic, and ionic stresses. And well, it's not clear if we really have an increase or if we really cannot deal with this increase so well. So these are the two possibilities. Either we get older and we have more oxidative stress, more metabolic stress, or more ionic stresses, or either as we are older, we cannot fix it. Our cells cannot fix it anymore. So the result, when you are young, you're going to repair, you're going to have a healthy individual. If you don't repair, you have accumulation of damage for proteins in the membranes, and you have a neurodegeneration. The neurons are much more sensitive to this type of stresses, and the neurons are the cells in which, with age, this gets worse and worse and worse. So the concept is the balance. I think I have a slide after. The balance between stress and repair gets broken with age. So, but the age is not the only factor. So in the aging brain, we're going to see oxidative stress, metabolic impairment, dysregulation of the homeostasis. So the homeostasis is not working so well. The homeostatic mechanisms are not working so well. So the cell is not only exposed to this narrow window of conditions, but also to, in some cases, not so narrow, and this is damaging for the cell. There is what we call protein aggregation. So you know what is protein aggregation? It's important for me that you guys talk, because I cannot know if you don't understand me, or if I'm going too low level, or if I talk in too much biology or too little. So it's very important that you guys, so you don't know what is protein aggregation, okay? Yeah, it's part of this class. So one of the main landmarks of neurodegeneration is the protein aggregation. And this happened because in the last, it's not the last century, it's the century, at the end of the 19th century, people start to live longer. So people start to have these diseases. And the physicians, when they open the brain of the people who die with neurodegenerations, could see stuff in the neurons. And these were like balls. And were balls of proteins, which were very insoluble. I mean, they were not soluble in the cell material. They were like a precipitate. It's like if you make a choco, you put too much, and it goes down, okay? So it's not soluble, number one. Why happened this with the protein? So when the protein, the protein has a structure. The protein is synthesized as a chain of amino acids, as a linear chain. But this is not the functional protein, no? The functional protein, in order to work, needs to make a structure which now can do things, okay? How do you believe this happened? How do you go from this structure to this structure?
Cutting it.
Eh?
Cutting it.
No cutting.
Yeah,
Yeah, so there is a forces that keep this, this is some sort of equilibrium between the two forms, But you can imagine if you have 1,000 amino acids, you have thousands, 100,000 ways to shape this protein with this cell. Indeed, this is one of the biggest mysteries in biology. We don't know how a protein chooses the right conformation. And we don't know, in many cases, how the protein, because even the protein doesn't fall like this, but now falls like this, let's say. And this protein maybe now not only cannot do the function, but for example, can bind to itself. It binds to itself, and this binds to itself, and suddenly all the ionic bonds are with itself, so cannot interact with the water or with the cytoplasm, so will precipitate and will go down. The typical example is the prion protein, the mad cow disease. What happened in this case, what these mad cows have is some protein which is incorrectly folded. And what is interesting about this protein will not be a problem by itself, but when you eat it, make your proteins to fall wrongly. So somehow can interact with the proteins, with some endogenous proteins, and make them fall wrongly. So you have also these aggregates of proteins that are not soluble, and you get into big trouble. So protein aggregation is a very important landmark in neurodegeneration. So you guys are following me? Yeah? Okay. So the other one is mitochondrial instability, reduced clearance of toxins, DNA damage, apoptosis. So also it's very difficult to say what is the cause and what is the consequence, you know, because basically when you have a cell or a brain which is subjected to so much stress, everything is going to go wrong. And these diseases, in the very terminal stages, everything goes wrong. And at the end, you have neuronal dysfunction and death. So you have many diseases in which the age is not the only factor, and in which you have specific mutations that favor the disease. So if you are unlucky, you can get this stuff, and there is very known a risk factor for Parkinson's disease when you have certain mutations in an enzyme called alpha-synuclein. It's not an enzyme, it's a protein. It's not an enzyme. Or ApoE4. And in many other diseases, you have many mutations. Also, the environment may play a role. It's like a very, it's very difficult to determine how much is the environment and how much is not. If we have time, we're going to talk today about this. And there is some factors that's supposed to enhance neurodegenerative diseases. On the other cases, there are some mutations that will help you to deal, and you will have less chance to develop these diseases. And for example, some diet and lifestyle can also help to not have some of these diseases. And I'm sure that Mona told you, because if you smoke, you have less chance of Parkinson. Okay, yes?
HD? I know AD and PD work HD.
Well, Huntington's disease. Yeah. So, yeah, the main two, or the main three neurodegenerative diseases, Alzheimer, Parkinson, and ALS, so multiple sclerosis. Okay. The rest are all genetics, like Huntington's disease and some of the ataxias. I don't think we're going to talk much about it. So when we look at people with the neurodegenerative diseases, we can see that the cellular damage is progressive and is cumulative. And the question is why? I think I said it before. Why is progressive and cumulative? What you would say is the main reason? No? No, yeah, but just let's think the properties of the neurons. Accumulates, you can divide it. So indeed, there are very nice experiments in cells. If you have cells, normal cells, skin cells, let's say, or that are dividing, the toxicity of some of these aggregates is much less than if the cells, we don't let them divide. And it's because this aggregate gets like having a tumor inside the cells, get bigger and bigger and bigger, and this, okay?
Sorry, just out of curiosity, can you tell us why smoking?
Indeed, I don't. I think Mona has an hypothesis about this, but I has.
She said it's only in a certain age. You have to be like 60 and older.
I guess if you survive until 60.
Yeah.
No, I know the study. There is a epidemiological study. There was a completely. They didn't check this specifically. They just found this, but
By chance,
may have to do with the nicotine receptor. I don't know, with something like this, but.
With cholinergic receptor.
Yeah, exactly. With the subtype of cholinergic receptor, the nicotinic receptor. So it may have to do. It probably has to do with this, but instead, agonists of acetylcholine are used in some treatments against, but I think it's against Alzheimer. I don't think it's against Parkinson, but may have to do with this. So neurons are also more susceptible to these stresses, and when they die, are not replaced. So why they are more susceptible? And because they use a lot of energy, because they have a lot of membrane, okay? And because they don't replace, because they don't divide, okay? This is, as I said, this is the general view. It's obvious that aged cells are much more sensitive than the new cells. Yes?
They do not divide, but they do have
Neurogenesis.
neurogenesis in the hippocampus, so we do have more.
It's not clear the function of this neurogenesis. Yeah. No, I agree with you.
So
This, I told you, this is the classical view, and I indeed not sure that it's also why. Yeah. But most of the neurons are not replaces. Okay, like as you're telling me, in some places in the olfactory bulb also there is neurogenesis. In some places there is neurogenesis, but it's not like happening in the liver or like happening in other tissues, which you really replace all the cells all the time.
Also in the peripheral nervous system, there is replacement.
Yeah.
Right?
Yeah. So also some other arguments are we have so many neurons that we use only a very small fraction of really them. So doesn't matter that indeed it's like saying that it's replaceable because we don't we can make a connection to another one. That's one of the arguments also that saying that this is not completely true. Yeah.
Why does the fact that the neurons need more energy or use more energy make them more susceptible to disease?
Yeah, because we are going to talk a little later, but basically the mitochondrial function, you need to deal with a lot of free electrons and oxidative substances, which can oxidize proteins, can oxidize DNA, provoking DNA damage and mutations, can oxidize mitochondria, so can damage the pathways that help to clear aggregates, for example. So if you use a lot of energy, this comes with a lot of cost. So also I should point out, I think I have it after in one of the slides, that the neurotransmitters are not a synthesis of neurotransmitters is something that involves the very toxic substance for the cells. All the intermediates in the synthesis of a neurotransmitter is very toxic. So this is something that also puts the cells in stress. Well, this is exactly like we were talking before. I just believe it was better to say before. The cells generally don't divide. There's some exceptions, so the damage is cumulative. They use a lot of energy, so they have a lot of oxidative stress. Most of them generate toxic compounds that neurotransmitter or intermediates on their synthesis lead to more oxidative stress. And they have a huge surface. The area-volume ratio is very big compared to almost any cell type. So why do the neurons die in neurodegenerative disease? I would say that it's mainly three. I would say this is, again, a very classical view of neurodegeneration. Three ways to see it. One is protein misfolding, or just the protein fold in a different way. And this can lead to aggregates and also to loss of function of the protein, both things. Oxidative stress and apoptosis. You know what apoptosis is? Apoptosis is programmed cell death. Yeah, so it's a program by which the body identifies a cell as dangerous for the organism, so just makes the cell to eliminate itself. The cell goes to, you can also call it like suicide, like a cell suicide. Okay, so I know you said, okay, let me, before going into this, say one more thing. So the classical view then seems very straightforward. These are the problems we need to deal. The proteins misfold, and the neurons have a lot of oxidative stress and a lot of surface. So what happens with age, that the cell gets old, gets less strong, the repair systems are not so good as before, and that's why all older cells are much more susceptible than younger cells. That's the interpretation. But why you believe that the older cells cannot deal with the stress anymore? What will be the why? I don't want any strange interpretation, just with complete common sense. Because if the young cell was so good in dealing with the stress, no? Then there shouldn't be any cumulative damage, because there was no damage. The young cell was taken out. What happened with the old cell?
You already accumulated damage.
But if this means that it's not so much about the old cell, but it's more about the young cell not doing the job well, no? That's what I mean. If the young cell, the repair system was good, shouldn't have any damage. Indeed, in young cells, you see very little or no damage at all. So something happened to the cell. Something happened to the cell in a way that the cell cannot repair the damage anymore. And there is, yeah.
Well, I mean, I think in some ways it makes sense because there are natural, normal, healthy changes that do happen in older adults in terms of memory loss or a different way of thinking that aren't inherently bad or wrong, but that might make the brain more susceptible to extreme or pathological forms of that.
Okay, this is a, okay. What you mean, this may not be a malfunction, but just like a maturation of the brain. Like the brain is in a different stage that as a subproduct is more susceptible. And my answer goes not far from there, yeah.
I think if the cell itself didn't change, then maybe its environment changed.
You mean that the body cannot now deal as the homeostasis around the cell?
But maybe the extracellular environment.
No, could be, but the same happens for the cells in the kidney, and it's not that you're going to have much more kidney failure. No, I don't know if I give a good example because maybe you do, but it's very clear when you take the difference in the neurons is really different with the other parts of the body, and could be, but I don't think it is. But it's a good possibility too. So what the possibility is emerging now in the field, sustained by some very elegant experiments in worms, indeed, first and in mice, is that maybe aging, the fact that we get old, is not a consequence of that our body cannot deal with the stress and we just use our hands and then the hands get old or used. It's like a program. Maybe it's just a program because we need the population to be replaced. The population needs to be replaced. So it's like a sort of a program. Indeed, in worms, you can stop this program, and the worms can live almost forever. Like it can live 2, 3, 4 times more than supposed to. And they have done some of these experiments in mice too. So is this idea that you can really, that the aging is a program, is not a consequence of injury or of use of the stuff. And I think that this is a fascinating hypothesis, especially because then we could live forever, no? But
It's not a problem, it's like it's the plan. I mean, that's
yeah, it's part of the plan. And as a species, this may give an advantage if you have it and if you don't have it, okay? So there is in some cases, because there should be a reason, and it's just going too far from the class, but I just want to say, for example, the eggs accumulate mutations over time, okay? So if you have, the older the woman who has kids has more chance to have mutation during the life, so you cannot have a person of 80 years having the kids, so better you cut it before and you have a program that get rid of the people who cannot reproduce. So let's say like this. And I say this in this class because there is, in the last 3-4 years, there was a few papers. And I think that the pioneer in this was Ehud Cohen, who is going to give a class here, in which he showed that if you play now with the aging program, you can get wolves or mice which can resist neurodegeneration even when they are old. So looks at the damage may not be cumulative. The damage may be very, very quick, but the damage only happens when you are old. That's the main thing, and it's part of the aging program. That's
and what is that you're speaking of a program? Where is it? I mean, what is it?
Yeah, it has to do with the production of insulin and some hormones. And if you eat, if you eat very little, if you do what you call calorie restriction, indeed the level of insulin change, because they know that the people who eat very little live longer. This is known. This is known that if you eat, I think it's 40% that you're supposed to be eating, or that you are going to eat if you have free food. If you eat 40% of that, you live much longer. And this is true for all the, what is it?
Does it have something to do with diabetes?
It may, but I don't know. Yeah, insulin obviously has, but I don't know.
No.
It's the same pathway, but I don't know.
People having diabetes to get neurodegenerative diseases.
That's a good question, and I don't know. Probably, because having diabetes brings you every problem, but no, but I don't know. Well, what happened here? No, it's got unplugged. Okay. So what I want to tell you that everything I'm telling you about the cumulative damage, this is the story that you will see from everyone. But there is in the field, there is a view now that is growing that it may not be the real story. And I'm trying to think, he's not coming this year, I think. But even in humans, some studies are showing that some of the aggregates in Alzheimer only form in the old people. It's not that they were in the young and are getting bigger and bigger and bigger and bigger, but that only form in the old people and very, 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 very quick there. Okay, so let's look just at the, but this is not the official story, this is a side story, okay? So let's look at the cell. The organs you need to remember here is the mitochondria, the lysosomes, and the endoplasmatic reticulum. Why? Because these are the places where a lot of the proteins are done, are degraded, and where most of the energy is produced, the mitochondria. So you know that a lot of proteins are done in the endoplasmatic reticulum, no? A lot of the proteins are done in the cytoplasm by ribosomes, but some of the proteins, more than half, are done in the endoplasmatic reticulus. And indeed, the lysosomes, we're not going to talk today. So why the neurons die in neurodegeneration disease? The pathway we are going to mainly see today is protein aggregation that is a consequence of protein misfolding. So a protein must be formed normally, okay, to function. So we have the genetic information in the DNA, which is transcribed, translated, and the last step is the folding of the chain of amino acids into a protein, and then you have a functional protein. I should say that in biology, the shape means function. You know, it's not that the way you fold means what you can bind and what you can do. Okay, you know the way an enzyme works. No, you know what is an enzyme? An enzyme is something that, for example, from A plus B can do C. No? If you take a crazy amount of A and a crazy amount of B, and you put them together, you are going to get some C. This is a natural reaction that is going to happen. It's not that the enzyme is doing something that it could not be done spontaneously. What the enzyme can do is make it happen very fast. Catalyze it, exactly. That's the real, that's the term. So then you can imagine in the protein, we have a place that can bind very well because the shape or the interaction that can establish with A are very strong, and this is B. And what the enzyme is just doing here is just putting these two guys next to each other, so they will do the reaction. So this is what I mean that a structure means function, and it's very delicate equilibrium from all the structures that this amino acid chain can form to get to this one. So any slight change on this is going not only to kill the enzyme, but maybe give new properties to the enzyme, like bind to itself or bind to something that shouldn't be binding. So it's going to be toxic. So folding is very, very important, proper folding. So if you have a linear amino acid change, and in order to fold, I told you this is one of the biggest challenges in molecular biology to understand how a protein folds. There is many theories. One theory suggests that the protein tries to fold in every single way and most of the ways are not stable until it finds the right or the more stable way. Other theories say that it's more complicated, and I'm sure it's more complicated because if you just calculate the time that this amino acid change will take it to try all the shapes, this is much more than what actually takes or the time that actually happens. So then I'm going to talk a little about quality control of proteins, and by quality control I will mean all those systems that assure that the protein doesn't miss the right folding, okay? Well, the first one is happen very quick, even when the protein has not been completely formed. And don't, so you guys, is fine this pace of the class, or is too fast or too slow? Okay. And don't get crazy now with the names and the stuff. I will tell you the concept, okay? So these are ribosomes. You know, the ribosomes are the machineries that made proteins out of RNA, an amino acid. This is the RNA. And you can imagine that if you have a long protein in this, for example, this part interacts with a lot of parts which are on the other side. Okay? So these two parts cannot interact until they are not synthesized. And of course, the beginning or the end terminal is synthesized way, way before the C terminal. So you need mechanisms that prevent, well, first, you need mechanisms that prevent these nascent peptides, the ones that are being made, not to interact with each other very quick before the whole protein has been made. And on the other hand, you need mechanisms that don't prevent one protein to interact with each other, because this is going to interfere even with translation, maybe. So that's a matter, and I think there is a slide that is just for your information if you are interested. What is the mechanism that prevent these intermolecular interactions, interaction between different molecules, or intramolecular interactions, interaction in the same molecules as the protein is being made. So there is a whole machinery to prevent this type of interactions. Okay? And it's based on what we call heat shock proteins. Doesn't matter. Okay? Just this is the first problem or folding problem that the protein needs to see or to deal with when it's being made. The second one, afterwards, is after you have the protein, you have what we call assisted folding. So as I told you before, if you calculate the time that this chain of amino acid needs to try all the conformations to find this one, this will take years, but the protein is folded in seconds, minutes. So this is achieved by, it's not years, you know, maybe it's hours, and you can do it in a tube for many proteins. This is achieved by the help of other proteins, which are called chaperones. I didn't put it there, but it's in the next. And you know, the chaperones help the proteins to fold. Once the protein is formed, we want the protein to keep the structure, okay? So we are going to have proteins that are going to help the proteins keep the structure and will prevent the formation of aggregates. And last, but very, very important in neurodegenerative disease, we have a machinery we can degrade the proteins that are beyond repair, that cannot be refolded, and the ones that are old also. Because this, and this, if I mix up the stuff in your head, just tell me. But this is very important. You know, people talk about old cells and new cells. But really, what is different in an old cell and in a new cell? Because the proteins of an old cell are new proteins. The proteins are being made all the time. Even some of the lipids, cholesterol in the membrane, everything is being made all the time and replaced all the time. The only thing that is not being replaced is the DNA, and you could say, okay, there is some mutations that are accumulate. This is the only argument that could be done, but the DNA in neurons, in many cases, because it's not dividing, it's not exposed to some other stresses, okay? But that's why I want to mean that the old cells don't have old proteins. The old cell has new protein. Okay, so this, this is the thing. This is chaperones. So basically, if you have a protein which is unfolded because it was just made, okay, or that has, for some reason, get a wrong conformation, then there are enzymes called chaperones that use ATP. ATP is like the
Fuel.
not more the fuel, like the money of the cell. You can say it's a fuel. It's the money or the way that the cell keep energy. Yes, fuel is a better word. Using this fuel, you can put it into native protein. And when the cell is subjected to stress, there will be many proteins that are going to go in this direction. So you will probably, many times, stress situations are going to lead to increase of chaperones in order to solve it. Okay? Chaperones are very, very effective. Now, some proteins cannot be refolded. Okay? So then they are target for degradation. And they are degraded by a machinery that is called proteasome. These two types of functions are like a folding when the protein is made and refolding when the protein, for some reason, got the wrong conformation after a while, is done by chaperones. So chaperones help protein folding in normal conditions, and also in stress conditions. I told you many stresses can make a protein that was correctly folded to unfold. So this is the same. If you have a protein in the folded state, this doesn't work now. Okay, if you have a protein in the folded state, the chaperone will take it to the native state, and this passes through some folding intermediate. Basically, it doesn't matter. I'm not going to. Okay. This is just one example of chaperone, the way it works. Somehow the chaperone can help the protein to find the right conformation. As I told you before, a protein needs to, hypothetically, needs to try hundreds or thousands of conformations. The chaperone has the ability to show to the cell a few conformations, and one of these conformations is going to be the most stable one. Okay. So what happens in neurodegeneration? Basically, when you have synthesis, you have an equilibrium with the folding, and now many stresses are going to move the equilibrium to misfolding and aggregates, and some chaperone will bring it back. Okay. This is basically saying the same thing a few times with different graphs for all of you to understand. It's clear? Everything is clear? All of you guys? Sure? Sure? No? Yes? Okay. So many of the chaperones have been identified as heat shock proteins. Well, it's there. But why you believe you can, at least you can read it, but why they were identified as heat shock proteins? Because these proteins are expressed under heat shock. So if you put cells at 42 degrees, for example, cells is to be at 37. So if you take a tumor and you put it at 42 degrees, so what happened? What happened? These proteins are expressed. And why is so important? What will happen at 42 degrees with the protein? Why we need to express these chaperones at 42 degrees? You understand? If you heat shock cells, if you put them in a heat in heat, like 42 degrees, for example, they are going to be expressed. What happens if you take a cell? What is the main danger for a cell when you increase temperature? Yeah, what will be the main damage you are going to do with high temperature?
Denaturation of the
Yeah.
protein.
Yeah, when you increase temperature, then you are going to denature proteins. So what the cell has a system to protect against that. So these heat shock proteins are expressed in response to high temperature, and this is that's the way they were discovered. So this is just some of the name of the chaperones, some HP 190. Some of them are very, very famous. Indeed, they were discovering bacteria first. So the last pathway we need to talk about when we talk about folding and aggregation is the degradation of the old or damaged proteins. So this is done by two systems, the UPS and the lysosomes. And I'm going to go through the UPS system, so don't kill yourself now. So this is the ubiquitin proteasome system. And I really don't want to go. You're going to kill me if I go really deep into this. I will tell you just the concept, okay? So the proteins which are, we have a strange folding, okay? Proteins which are, we don't have a good folding. Somehow they are identified, okay? They are identified and they are marked with a molecule which is called ubiquitin. So and it's a very complex pathway, okay? But I will show you here. So because I give the same class only for biologists, and that's this we see it there well. But the proteins which are in a state which doesn't seem very good are tagged, marked with an ubiquitin. It's a very small molecule. Then this protein is tested again to see if it really was not well folded, and if it was okay, the ubiquitin is removed. But if it's not okay, then it's polyubiquitinated. So this means they have many ubiquitins. So when the protein is not in good shape, it's going to have many ubiquitins, and now it's going to be degraded by the proteasome. So the proteasome doesn't degrade any protein, only degrades the proteins which are polyubiquitinated. The proteins which have a lot of ubiquitins are the ones that are degraded. This is a tag for degradation. So basically, how we can think about it, basically this unfolded protein is going through the cell. The proteasome is only in a very few places in the cell. So you cannot have the proteasome going to everywhere and degrading everything that is not good. So you have like other proteins like policemen that are putting a mark there. And then the protein keeps going up and down all around the cell, and another one will put the mark. When you have many marks, when you pass near the proteasome, you're going to be identified and degraded. That's the logic of the system. Okay. Now, the proteins, this is the way that you can degrade proteins that are in the cytoplasm, that are freely moving the cytoplasm. The proteins that are in the membrane are degraded by the lysosomes, okay? And basically, you have this is very common in receptors, so they can be internalized and degraded in lysosomes. That's what you need to know. So protein degradation is very important because the synapses are very dynamic structures. They keep changing all the time. And you probably listened or heard the term synaptic plasticity. And this is the property of the synapse to change connection from one neuron to the other neuron, to enforce connection with one neuron with other neuron. And all this is done by virtue of protein degradation and protein synthesis. So you need to make new proteins and you need to degrade other proteins, depending on the type of a connection you want to establish. So doesn't matter here the details. But it's not only that you want the degradation system for proteins to work well in general, but especially in the neurons, which are all the time remodeling the synapses. You need a very, in order for the neurons to work well, the degradation pathways need to work well, okay? So, and this is the same, proper protein degradation is key for synaptic homeostasis and for synaptic plasticity and for synaptic remodeling and for synaptic function, okay? And I think when we talk about Alzheimer, we are going to talk about this, okay? Okay, any question?
Yes. Can you explain a little bit about the way of degradation of the neurotransmitters by the transporters, you know, that takes them back to the cell?
Yeah, this is what I put. There's the lysosomes. Okay, that's what I put here. I don't want to go into the detail here, but basically the receptors usually are internalized together with the neurotransmitter or with the ligand to it, and they are degraded in a structure that are called endosomes. Okay, basically you have bags of factors which have enzymes that can degrade everything. These are called lysosomes. So this thing is internalized and is fusing with the lysosome, and everything is degraded. Okay.
And after degradation, being rebuilt.
Yeah, the lysosome is intact, but this is it's synthesized again. This is part of what I'm saying. You have all the time synthesis and degradation of stuff. The membrane of the neurons is a very how to say, dynamic structure. So I want to ask you, if you want to text message, just go out a second, because I feel it's like a little. Okay, problem. Okay. So this is the first thing I want to say. That looks like this is a fabulous system, no? We have a system that is amazing. Can control at the moment the protein is done whether there is a problem. If we have can help the protein to fold properly. If we have proteins that are not folding well, can help them to refold. And we can degrade all undamaged proteins. This system is fabulous. So we are really amazing. And this is usually, you know, how to say, I don't want to attack physicians. That's when you describe a system like this without thinking in the possible problems, it's like many times the traditional view of medicine, no? And this looks beautiful. However, if you just tip a little there, and where there is a little of stress, you get into trouble. That's the key thing. And as I told you before, I hope I convince you, the neurons are really prone to stress. So still, the cell has many responses to stress. So this is just an example with heat stress, but this happens the same with ionic stress, any type of stress. You basically, the first things that the stress is doing is somehow affecting protein folding. Okay? Most of the stresses, directly or indirectly, are going to be touching protein folding. And this is very, very important for the cell because the proteins are doing everything in the cell, are the workers on the cells, and also because the proteins are in the verge of aggregation, let's say, okay? So they are very sensitive to difference in folding because this can bring the proteins to aggregate together.
This is also correct to toxicity stress?
Yeah, many toxic stress affect protein. Well, in many cases you may have DNA damage, no, of course, UV, or you have many cases you have also mutations in the DNA. But most of the most dangerous thing for the cell is to have damage in the protein folding. It's the most, I would say, the most sensitive. It's the most sensitive thing. Okay. When you have problems with folding, the first thing that happens is that you have attenuation of protein synthesis. So the cell tries to stop making more proteins. Okay. Because as you know, if you have aggregation of a few proteins, if you have more proteins and more protein and more protein, it's going to do a ball and a ball and a ball. So the first thing that the cell is doing is stopping synthesis of protein. And this is one reason. The second reason is that you really don't want to invest energy in anything else except for dealing with the stress. So you don't want to make more protein and invest energy in doing more protein. Other very common response is to promote degradation. So you try to degrade especially the aggregates of the proteins which are misfolded. And the third response is that you try the same stress or the protein folding problem can be sensed by the cell, and this can induce the expression of mainly chaperones, okay? And these chaperones are usually called heat shock proteins only because were discovered happening as a consequence of heat shock, not because they only help the cell to deal with the heat shock. Okay, don't worry about this. So basically, this is to tell you that in many times, and this is I think from the biological point of view, is very, very interesting. It's not that the cell is sensing the stressor, I mean the heat or the heavy metal or whatever is being the stress to the cell, but the cell, what is sensing is the damage or the problem in the protein folding. So the cell can sense problems in protein folding. And this is very important because the cell never knows which type of stress can be exposed in the future. So if you have a system that can also sense the folding problem, the consequence of the stress, then it's much better. Okay. I think one of our guest lecturers will talk about this. Okay, so the class is until 4, no? I'm correct? Okay. So what happens when the quality control fails? You have aggregation. And aggregation is, like as I said, is the landmark of a neurodegenerative diseases. And this is the story, the official story again, because there's some theory that they propose that aggregation is a protective mechanism. And there is very, very good evidence for this, but still, big aggregates are protective, but the small aggregates are very, very, very toxic. So this is a common view, that the small aggregates are very toxic and the big are. But we are going through the story, and you're in the next class, probably we are going to talk a little more about this. So aggregation then is a failure on the quality control. Why? Because proteins that are misfolded supposed to be either degraded or either reshaped to the native fold. So aggregation only happens when the quality control fails. And what happened? What people observe in the aggregations is that these aggregations are polyubiquitinated. You remember what is ubiquity? What is? It's the mark for degradation. So the cells identify these guys as a problem and it's marking, but somehow the proteasome cannot degrade it. That's one of the key things of the aggregates. The proteasome can't degrade the aggregates. Okay? Why? So because of the structure? This is one possibility because of the structure. The structure, some people say that number one, that the structure makes them unbreakable. And indeed, even if you want to break it biochemically, it's very, very difficult. Number two, they say because the structure is like this, not only the proteasome cannot break them, but when the proteasome tries to break them, they get stuck there. And then the proteasome doesn't work anymore. So then you have even more aggregation and more aggregation because even the proteins that you will be able to degrade, you're not degrading. So this is a thing. And this is what I told you that at the beginning of end of the 19th, beginning of the 20th century, people open brains of people with Alzheimer or Huntington or Parkinson, and they found these huge aggregates in the cells which are insoluble and which can be stained with Congo red or many common dyes that people use when doing histology. And this was the main thing that you could see in the brain when you don't know anything about molecular biology is this. So you could see, and again, the common view is that there is a balance between the misfolding protein and the folding capacity. And the idea is that when you are young, you are okay because the folding capacity is more than the misfolding proteins, or maybe not, but the misfolding protein didn't accumulate enough to be a trouble. But as you age, you go unhappily to the other end. Okay, so there is a lot of human diseases, not only neurodegeneration, which are associated with the formation of aggregates, okay? And this even like Alzheimer, some other neurodegenerative disease, but even some kidney diseases, even some type of diabetes, also are associated with this, what we call aggregates, okay? Some of these aggregates, in this case, are extracellular, but can also be intracellular. Okay. It doesn't matter, okay. I want to, okay. So some of the common features of protein misfolding in a neurodegenerative disease are the following. So this type of structures are called amyloid-like. They have a lot of resistance to proteins and detergents, and this means they are almost unbreakable. And they aggregate, doesn't matter what is the cross beta sheet structure, but you can imagine that if you have a protein which has a structure like this, the other protein goes like in the other direction and it's combined to it. They have a structure that make them polymerize one after the other. So they make a huge, I think I have a huge structure like this that go together many proteins together. So they become a huge conglomerate that aggregates, okay? Okay. I keep it because I'm going to forget this thing and I don't think I need it now. Thank you. So what is the relationship between the misfolding and the neuronal death? So somehow you have misfolding, then we know who knows how you get to cellular dysfunction, and then you know to neurodegeneration. The problem is that it's very difficult to study this, because if you take a brain of a person who died of neurodegeneration, then you are, whatever you are seeing here is the consequence of all this. So you really don't know if the difference with respect to a normal brain is the cause of neurodegeneration or the consequence of neurodegeneration. So, and if a person which was at the beginning of neurodegeneration dies and you look at the brain, you believe this person is normal because didn't have symptoms yet.
But that's why we conduct experiments on mice, for example, or
Yeah.
what the cause and what the infection.
Yeah, but it's still I think if I have time, I'm going to talk about this. There is a lot of, logically, this type of experiments also are, you are in danger of making a fallacy of affirmation of consequence. Because if you have 10 things change here, okay, and this gives you the disease, okay? When you have 10, let's say 10 different things that you can say about a brain neurodegenerative against a normal one. And now you make a mice which has one by one all of those things. Well, of course, the mice is going to, if let's say, if a lot of the things that are consequences of neurodegeneration that came downstream, because the cells is very sick, the cell go to apoptosis, let's say. Okay, they go to suicidal, the neurons die. If you make the neurons of the mice die, you're also going to have neurodegeneration. But it was not the first cause of the neurodegeneration. This was the last, one of the last steps. So it's also very, I work myself in my lab with animal models, so I believe this is the best approach we can do. But it's still very difficult because the main research needs to be done in humans. But for the molecular or for the most biological, indeed, flies and worms and excellent models, probably even better than mice or rat. But mice or rat are also very good. Okay. But the point here, you always need to go at the end to the human. Okay. Sorry for this. No? Any question? No. Okay, now it's going to work, I think.
I think it's again the wire.
On the wire is put it. Okay, I think now. Yeah. No? Yeah. Yeah. Okay. No. Nice. Okay. So what is the role of the, because I told you there is a system, the proteasome, that will degrade the aggregates, will clear basically the program. So this is a little what I told you before. What is the role of the proteasome and the UPS response in neurodegeneration? And the UPS is this lysosomal response I told you that can degrade the proteins in the membrane. So we have this system to deal with the unfolded proteins or with the aggregates. Why? What is the role of this? So I told you that the aggregate can inhibit, can choke the proteasome, and this also can lead to cell death. It also could be possible that what happens in neurodegeneration is that the proteasome is dying for something completely unknown for us, but this is very central to neurodegeneration. And now you have the aggregates, but the aggregates may be a consequence of a main dysfunction of the proteasome. The other possibility is the aggregates are protective, and there is no relationship between the proteasome dysfunction and the aggregate. The aggregate is like a program of the cells to protect. And I believe that there is evidence to support every single one of these hypotheses. So probably it's a little of each one, and it may vary a little regarding the disease. Okay, so in the rest part of in the last 10 minutes we have here, I'm going to try to talk a little about oxidative stress. So about apoptosis, I want you to know that apoptosis involves, the mechanisms are are quite complicated, so I don't want to go doesn't matter to go into detail on this, it's not important. It's important, but not in this class. Okay. So you know what the free radicals are? What you need to remember about free radicals, well, it doesn't matter, they have an unpaired electron in their outer orbit, and this makes them very, very reactive. Exactly. So these free radicals are ready to react with whatever cross in the way. And the free radicals can chain reaction, because what they're going to do with this electron in the orbit, they're going to take it from someone else, and this someone else is going to take it from someone else. So once you have one free radical in the cell, it's going to do a disaster, because it's going to amplify the signal. And these are generated by high energy radiation, also by oxidation. And this is, you know, the cells is doing oxidation all the time when it's producing energy. So the fact that the mitochondria is producing so much energy means the mitochondria is also generating a lot of free radicals which are highly reactive. And this is just a way you can get a free radical from the outside by chemical agents, by inflammation or microbial killing, by irradiation, by oxygen, and by age-related changes. So the classic definition of oxidative stress is that it is the production of reactive oxygen in excess of antioxidant mechanism. So it's a balance between oxidative and oxidative if it's more. So the modern definition is you talk is the same thing with the nicer words, but there is an altered homeostatic balance. Like the homeostatic balance reference of the capacity of the system to deal with this oxidative stress. So the balance doesn't exist anymore, and as a result, you have more oxidation than antioxidation activity, and you have this, okay? Doesn't matter. So this is okay. Let's skip this. What they want to keep in mind that in the core of the production of ATP, in the core of the production of energy, there is transfers of free radicals. Doesn't matter the coenzyme. Doesn't matter why, but the free radicals are in the core of the mechanism of production, of normal production of energy. Okay, it's the same. So when you have free radicals and you don't have a mechanism to protect or to antioxidant mechanism, you have a lot of modifications in all the type of normal or common macromolecules. So you're going to see lipid peroxidation, and this is something that you can see in Alzheimer. So the lipids also get oxidized. You're going to see protein oxidation. Also, you see it in Alzheimer. You're going to see nucleic acid oxidation, and you're going to see a glycosylation, that is oxidation of a sugar-like molecules. Okay, so basically, at least in very advanced stage of Alzheimer and most neurodegenerative disease, you have a lot of oxidative stress. Okay, and this affects all the macromolecules in the cell. But you cannot say that because of the lipid peroxidation you have Alzheimer. This is clearly a consequence of the oxidative stress. Okay. Yeah, that doesn't matter. So the mechanism of free radical injury, basically, if your neurons now cannot deal with oxidative stress anymore, you're going to generate free radicals that you cannot get rid, and this is going to oxidate lipids, okay, fat macromolecules, which is going to make a damage to the membranes. And the integrity of the membranes is very, very important for many things. First, to maintain a barrier between the intracellular and the extracellular media. And also, for example, the membrane are very, very important for generating energy. Okay? And it doesn't matter here, but in respiration, this is very, very important to have a membranes which are completely separate. You also have proteins in proteins that cross-link and fragment, and also in DNA that can lead to mutation. But this is quite secondary, the effect on the molecules, and is related to the last stage, which is the death of the neuron. So in this type of oxidation of lipids was recognized many, many years ago, because people found that physicians found when looking at brains of people with Alzheimer that they have a very oxidized lipid called lipofuscin, which is finely granular, and they can really see it in the brain of people with the advanced Alzheimer. Is this thing, which is yellow here, it's a neuron, this here, but so it's very easy to distinguish these oxidized lipids in the, so this is a hallmark of Alzheimer's disease and many neurodegenerative disease to have oxidations of micromolecules. The more visible one is lipids. Okay, so basically, and we're going to finish here. You have a balance between a source of oxidative stress and natural or artificial source of antioxidant. And depending on this balance, you're going to get more or less oxidative stress. And the view is that this oxidative damage is central for the beginning of this disease, but also for the end, because this is at the end what is going to damage the proteins and lipids, etc., etc. Okay, so I think we're going to stop it here. And if I remember correctly, I'm also with you next week, if I remember correctly. Okay? Okay, good.